Okay, thank you. Good afternoon, everyone. First of all, I would like to thanks to the all committee who organized this wonderful symposium. And I am relatively newcomers in this area, especially for the computer and drug discovery. And I'm so much excited today to be included in this symposium. So today I am Hydrolytic from Tianjin University, would like to deliver my talk entitled Virtual Screenings of Plants' Natural Compounds targeting the hydrolytic and lignin degrading enzymes of Ganoderma boninense. So this project was motivated by basal stem root is a major disease that poses a serious threat to the oil palm plantation in Southeast Asian country, especially for Indonesia, that caused by Ganoderma boninense. So during infections, these fungi produce the lignin degrading enzymes such as like lacase, lignin peroxidase and manganese peroxidase, and hydrolytic enzymes such as like cellulase, amylase, and silanase to target the major component of the cell walls in the oil palms. Therefore, inhibiting or the blocking both these enzymes could be a desirable strategy to control the growth of Ganoderma boninense. In this context, several measurements have been adopted to control the growth of the Ganoderma boninense, for example, metal ion and fungicide. Even though they are costly effective, however, they pose the higher toxicity against the human cells and environments. Therefore, non-toxic molecule and stables which have potential activity to control the growth of Ganoderma could be further investigated. By taking advantage of the in silico approach, here we used structure-based virtual screening to screen plants' natural compounds obtained from the online libraries and collected as the compound libraries. So that's the psychochemical properties after the docking analysis, the psychochemical properties and then toxicity predictions and biological activity of the selected compounds were then investigated. And finally, the top hits generated compounds, we further screens based on their binding interactions against the lignin and hydrolytic enzymes. And finally, we visualize the specific interactions using, using PyMol. We have initially selected the top 10 selected compounds based on their desirable binding affinities. And interestingly, here's the several elucidated compounds have a better docking scores than the methoxams as the fungicides against Ganoderma boninense. And this result further suggests that each selected compounds have significant binding affinities against lignin and hydrolytic enzymes. Then the psychochemical characterizations of the top 10 selected compound were further calculated and predicted using Marvin's sketch. And according to the agrochemical analysis, there are 8 parameters that can be used to exploit to further use to screen the selected compounds. And these parameters eliminates the compounds which are unlikely to be the chemical candidate as highlighted in the red colors. The eligibility of these parameters actually does not automatically qualify the specific compound as the Ganoderma inhibitors. However, it simply provides information on agrochemical likeness and help elimination for a weak ligand in the very early stage. Then the compounds which have the good psychochemical properties were further predicted for their toxicity predictions. And in this study, several toxicity parameters were exploited as mentioned in this slides. For example, MS toxicity and hepatotoxicity. And the compounds which exceeds the toxicity, again, these parameters will be eliminated, and then the remaining compounds were considered as the potential candidate as the Ganoderma inhibitors. And then finally, from the toxicity predictions, we selected top 2 selected compounds, and we further predicted their biological activities. So here's based on their biological activities that several compounds have the multiple target multiple target activities against the target proteins. And as shown in the figures that the PA score is higher than the 0.3 is indicating that have the potential pharmacological activity and biological properties as mentioned in the research goal. And then finally, this top 2 selected compound was evaluated based on their specific binding interactions against lignin and hydrolytic enzymes. We found the different binding interactions between the top 2 selected compounds and methoxam as the fungicide against Ganoderma boninense. And in general, that the ligand protein interaction were highly stabilized by hydrogen bonding and hydrophobic interactions. The more hydrogen bondings, the higher binding efficiency and inhibition. And the hydrophobic bonding were responsible for the target binding complex interactions. And from this finding, we can conclude several points. And the most important thing here is that the several compounds such as like epigenins, dedzane, and hesperidin are predicted to have the multiple target inhibitors against lignin and hydrolytic enzymes. However, in vitro and in vivo validation for this finding, such as like structure elucidation from the apo structure and the complex structure between the proteins and the ligand should be further verified and should be further confirmed. So this is the end of my presentation. Thank you for your attention. I open the stage for Q&A.
Thank you. We have several questions. So the first question from Kunal Roy: can you compare chemical space of plant products with that of synthetic compounds?
Wait. So the first thing, actually here for the plants natural compounds that we are collected from the online database, here's that the first ones we should like know that compounds are naturally coming from the plants, and there was no like a scientific scientific compounds that's coming from that specific compound. So actually to compare this, actually we can find this directly on the online database for that, which is it also indicates that this is really is naturally from the plants, or this is already synthetic from, or this is have some modification or the synthetic components of the plants.
Thank you. The next question from Timur Majidov: which criteria were used for elimination of chemical compounds based on their physical-chemical properties?
Okay.
Are there any rules like Lipinski for plants?
Okay, yeah. So this is really interesting questions. Just like the first one, we screens the plant's natural compound itself based on the docking scores. After we got the top 10 selected compounds here, then we use the agrochemical likeness. As I mentioned, that there are about eight chemical agrochemical likeness. It's a bit different with the Lipinski because this is for agrochemicals, but in general they are similar, but there are several parameters that used especially to screen the chemicals. And after we selected several compounds that meets these 8 parameters, then we further screen for their toxicity predictions, which is in these toxicity predictions we use PKCSM. So after we use the step of the further screening, then we only selected the top 2 selected compounds, which is we then finally evaluates their binding interaction and visualizes in PyMol. That's it.
Thank you. The next question from Vladimir Porokov: you mentioned usage of PASS program. How do you interpret these results?
Yeah, that's correct. That I use the PASS programs to interpret this biological activity of these compounds. So as I mentioned, that the compounds which does not exceed the toxicity value. So we found that for each target proteins, we selected top 2 selected compound, which is this top 2 selected compound we then finally to predict the past. And here is shown the figures that we use the PS scores to interpret, which have higher, so it have the biological properties as mentioned in the research goal. Thank you.